Hello, good afternoon, and welcome to all of our guests. My name is Chris Wigley. I'm the Chief Executive at Genomics England. And during the crisis, I also was seconded to NHSX as the Senior Responsible Officer for COVID data. None of us ever quite figured out what that meant, but it was a very interesting time. And it's my pleasure to be joined today by three eminent speakers to discuss this question of how can multi-omics data help us to respond to and be better prepared for future health crises. Professor Dame Sue Hill, Professor Albert Teneza, and Dr. Ewan Harrison. I'll ask each of them to introduce themselves briefly now. I'll then say a couple of words of framing, and we'll then have a presentation from each of our speakers, and we'll then open it up to questions. So we've already had a few questions coming in on the Q&A system. Do feel free to add to that as we go through. But first, let me ask our speakers to introduce themselves. Sue, why don't we go to you first? Sue Hill.
Thank you very much, Chris. I'm Sue Hill, the Chief Scientific Officer for England. I'm based in NHS England and Improvement and work across the health and care system. I'm the NHS SRO for genomics, but I've also been working as the CSO for England in NHS Test and Trace.
Thanks, Sue. And then Albert.
Hello, my name is Albert Teneza. I am a professor of quantitative genetics at the University of Edinburgh, and we work to try to understand how genetics drives differences between people in disease risk.
Thank you. And last but not least, Ewan.
Hi, I'm Ewan Harrison. I'm a HDR UK Fellow at the Wellcome Sanger Institute and the University of Cambridge, but for the last 16 months, I've been involved in the COVID-19 Genomics Consortium, COG UK.
Fantastic, thanks. And now that we've gone live, just a quick note, Albert, you're quite loud. Ewan, you're quite quiet. Sue, you're somewhere in the middle. So let's try and calibrate ourselves. And if I'm either loud or quiet, please give me the feedback as well. So I mentioned that we're talking today about genomics and multi-omics and the relationship with those and these kind of health crises like the pandemic that we're just out of. I'm conscious that in the audience here, there are some names I recognize and know are deeply expert, indeed far more expert than me in these topics. Equally, I'm conscious that we may have some people who are interested in these areas, but less expert, which is also great. So I thought it might be useful just up front to say, when we talk about multi-omics, what are we talking about? If we go back to the central dogma of biology, our DNA encodes RNA, which creates proteins, and those proteins kind of do everything else. They make our body. And when we think about DNA, all of our DNA data is collected together in the genome, and so we talk about genomics. When we think about RNA, we're talking about transcriptomics. When we're thinking about protein, we're talking about proteomics. And for other parts of the body, there's this area of metabolomics, which we often measure using blood plasma, but there are other ways of looking at metabolomics as well. So when we wrap all of these things together, that's multi-omics. And it's been notable and slightly incredible to me over the last year and a half as we've gone through these extraordinary times, how people, friends of mine, my family members who used to only have the vaguest or understanding and so on, to the virus itself, the COVID-19 virus, and to humans, or indeed other animals. So we're going to be hearing about sequencing the genomes of the virus. We're going to be hearing about sequencing the genomes of humans, and we're going to be hearing about some other tools, such as vaccine development, that are sort of underpinned by these technologies as well. To be useful, this kind of multi-omics data needs to be linked to all sorts of other data sets, whether that's logistics data sets for things like test and trace, whether it's how we analyze and then report the different variants on the viral sequencing front, whether that's clinical and health record data when we're looking at human multi-omic data, all of which raises great questions about trust, transparency, trustworthiness, and so on. And we've seen lots of examples through the crisis about how these data sets, these technologies can be applied at speed and at scale to really solve important problems. So hopefully that gives us a little bit of context. And at this point, I'll hand over to Sue, who is going to give us a bit of a tour d'horizon, so to speak, before we go to Albert, who will talk about the human genome sequencing and what we can understand about differential human responses, and Ewan, who will go a bit deeper on the viral sequencing and the CogUK response. So Sue, over to you.
Thank you very much, Chris. And if I could have my first slide, and I'd like to start by just thanking HRD UK and Genomics England for the invitation to speak this afternoon. And I'm going to rattle through this quite quickly. So where we were at the start of the pandemic is a history of investment and commitment to genomics, both human and pathogen, and established infrastructure, whether that was in the UK public health agencies, within the Genomic Medicine Service in England, within Genomics England, or linked to UK Biobank and a number of other academic and research organisations, and a key role for HRD UK. But between the Genomic Medicine Service and Genomics England, an important link with healthcare records, so a longitudinal health record. What we knew at the very beginning was the importance of having a sample framework, to have sample tracking, to look at sample quality, to have metadata to underpin sequencing, the reporting of genomics findings in a standardized way, agreeing nomenclature for mutations, whether that's human or viral, and the sharing of data both nationally and internationally. There was an infrastructure to support sample collection. We had engagement of pathology and genomic laboratories and pathogen genomic laboratories in PHE and across the UK. And we had an ability to expand existing services to cover sort of COVID-19 presentations, for example, through expansion of the age range for people in ITUs under the age of 40 who may have had an immunological underpinning suspicion to their COVID-19 response. If I'm going to go to the next slide just said, how did genomics inform our response? Well, it enabled us to rapidly implement molecular assays to detect the virus, and it also enabled us to establish new laboratories to make sure we were able to test at the level that was appropriate for the population. This was underpinned by a validation and verification process in place, but also our ability to have and to underpin this with end-to-end data and informatics developments, and I can't underestimate that. CogUK was established, funded by UKRI, and towards the end through DHSC and Test and Trace, and it is continuing with some of that funding. There was 20 sites started sequencing in early 2020, and there was an analytical tool developed that was called CLIMB for analysis of viral sequencing, but I know Ewan will cover this in a lot more detail. This was very much focused on NHS sample sequencing. In 2021, the responsibility for sequencing transferred to Test and Trace, so funded by DHSC. There's ongoing increases in capacity and in improvements in analytics being introduced, particularly for early variant calling, but right through to being able to get a quality assured sequence outcome that can be shared internationally in databases such as Gizzard. There is now sequencing of the virus from all testing sites, so those established under NHS Test and Trace facing the population, those in the NHS facing patients, and to support national clinical studies to identify new variants and viral lineages. And through this infrastructure, we've been able to quickly introduce targeted viral genotyping to identify common variants of concern, but in parallel to undertake full viral sequencing to know what is emerging in terms of initially variants of unknown significance that become variants of concern. But I can't stress enough to do this, there were a number of challenges around the end-to-end data and informatics developments. If I go to my next slide. My next slide just outlines what we achieved. So a world-leading sequencing capability and capacity, with the UK being the first to identify variants of concern, an extensive database of whole genome sequencing from individuals with varying response to COVID-19. So that's both on the human side and on the virus side as part of the genomics study. And with particularly COG UK, but now supplemented by others, with over 50% of the world's COVID-19 sequencing being undertaken in the UK and populating that global database for comparisons, but also to identify what's happening across the world. There's ongoing support for other countries in sequencing and interpretation of genomics data. And as Chris has said, there's been an increased awareness of genomics and its potential for healthcare, but the importance really of having data and data that needs to be shared to really understand what's happening at a population level, but also in our healthcare level. But the importance of both looking at human and viral genomes and the importance of the access to people's records. This is all informing future preparedness and more comprehensive pathogen genomics provision as we move forward. And my final slide, Chris, just outlines, if I may have that one. I think it's coming. Lessons we've learned, the importance of the interface between research and health and population care, the knowledge of how we can set up new tests quickly and use reference materials, some of the challenges of establishing end-to-end data collection, sample tracking, and robust analytics with QA as part of the QA process for genomes. There's still work to be done on how we interface between human and viral genomics, but ways of reporting results on a mass population basis so the results get delivered to individuals. The understanding of the use of data and what that means for public health and clinical intervention. How we can utilise and increase awareness of genomics to better embed changes in mainstream healthcare. That's both linked to infectious diseases, but for example, linked to rare disease and cancer. And really how important partnerships and national and international collaboration are, including through the two organisations I put on the top slide. So thank you very much. My last slide just outlines some of the contact details. Back to you, Chris.
Super. Thank you, Sue. And I'd heard that my internet was breaking up a bit in my intro, so I've tried moving to a new location. Hopefully that helps. Sue, well done on timing there. That was spot on. Thanks for the very insightful framing. I'll hand over now to Albert to tell us a bit more about the genomics of mortality and critical care, and what we've learned from that about how human genome sequences can help us understand what's going on. Albert, we can see your slides. You may need to flip to presenter mode, but they're coming through.
Is it in presenter mode now or not? No, okay.
I don't think so. if you click on slideshow at the top, and then that should then give you the option to kick it off.
I'll start. Usually,
that looked promising.
so you cannot see it.
We can see the slides. They're in, I guess, sort of slide making mode. But I think the
And like that.
if you just click on slideshow at the top, just next to animations.
Okay.
There we go. Bingo.
All
Got
right.
you.
Okay. Thank you very much.
Professor Albert Sneijder, all yours.
Okay, thank you very much. Good afternoon. So, as everybody knows, COVID caught all of us somehow unprepared, and we have had to learn how to deal not only with a global pandemic, but with new ways of doing things. And the work that I'm going to present is part of this. We have almost had to learn as we moved along how to do things differently. So COVID is a new disease and has two biological components. The first component is susceptibility to infection. That is why it is that some people that are exposed to the virus, they develop the disease, and some people that have been exposed to the virus as well, they don't develop the disease. And one can see this kind of example when it's actually at home, where it's actually very difficult to self-isolate, but some develop the disease and others don't. The other aspect of the disease is probably the most striking part of the disease is actually the wide range of variation in the symptoms and the severity of the symptoms that people develop. So whilst the majority of people will develop only minor symptoms, about 1% of the population become critically ill and need to be hospitalized, and many admitted into intensive care units. The other thing that we know is that for those that were hospitalized, we really need know very well how to treat them, and this is an example of this. So in this graph, the recovery study showed that if you treated people with glucocorticoids, there was only an advantage to people that have been receiving oxygen or mechanical ventilation, and there was no benefit to people that had not received oxygen treatment. So how could we actually try to find out new treatments, new drugs that could help improve the health of these people? And it is here where genetics actually can help do that. We knew in a study from a study in 2015 that genetically supported targets can actually increase or double the success rate of drug clinical development. And this was especially important in this case where there was quite a lot of urgency in identifying new drugs that could be treated, could be used to treat patients, or better still, to try to identify drugs that have been licensed to treat other diseases that could be repurposed to treat COVID. And that was the main purpose of our study here. And that's what I was going to talk to you. So the first thing that we did was to compare the genomes of 2,244 patients that had been admitted in 208 intensive care units across the UK, and we compared their genomes to genomes of UK Biobank participants at a ratio of 1:5, and the reason why we do that is to increase statistical power, that is to increase the chances of detecting parts of the genomes that are linked to the disease. To select those participants in UK Biobank, we actually selected them to be matched by ancestry, and then we adjusted the analysis. We took into account in the analysis differences in age, sex, and socioeconomic status. So what happens after one does this is that obtains a figure like this, and I'll show you. I'll tell you what this figure is. So on the horizontal axis we have the position of each genetic marker that we tested, and on the vertical axis we have the strength of statistical evidence. So everything that is above the red line we consider that is linked to the disease, and as you can see here that we have mirrored images. The reason why we have mirrored images is that on the top we have only data from genomic, and on the bottom we have combined data from the genome from an international consortium called the host genome the host genome COVID study, and also 23andMe data. And as you can see, that the strength of evidence increases as you have more data. So if you look at this place in the genome, this peak is at a lower level than this, and this shows how important it is to collect data. So what we found was 5 places in the genome that were linked to the disease, to critical illness. This one here on chromosome 3 had been previously reported, and the other 4 had not been reported. Of those 4, we used a technique called Mendelian randomization, where you combine data from gene expression in different tissues and genetic data from the genome from these patients, and we found that decreased levels of the interferon alpha receptor 2 gene were associated with increased level of critical illness, and increased level of the tyrosine kinase 2 gene were also associated with critical illness. So these 2 genes were obvious drug targets to treat the disease. So the IFN is involved in the interferon response, which is the first line of response to viral infection. And the other one, tyrosine kinase 2, it's related to the inflammatory response of the body to infection. We knew from previous studies, from clinical studies, that treatment with interferon in intensive care in hospitalized patients didn't improve their outcomes. So we believe that this is related to a pathway that is expressed early in infection and not when people get critically ill. However, the second one, the tyrosine kinase 2, is the target of a class of drugs called JAK inhibitors, and there are drugs to treat to block that pathway. One of them is baricitinib, and baricitinib is usually used to treat rheumatoid arthritis, and we hypothesized that would potentially be a good drug to use to treat COVID patients. And actually, a few months later, there was this paper in the New England Journal of Medicine where they actually show that treatment with baricitinib improved the outcome of patients that had been treated. So we think that this validates our approach, and this approach could be used not only to treat COVID, but to identify new drugs to treat COVID, but also to extend it to other diseases. And finally, I would like to thank all the people that have done the work, and especially Kenny Bailey, who was able to pull together a large number of clinicians and people working on this project, and of course to the patients and their family that helped with the study. Thank you.
Fantastic. Thanks so much, Albert, for that. Really another example of how things that used to take many years could be done at very high speed in the crisis around recruiting patients, sequencing the genomes, identifying regions of interest, using that to identify potential repurposable drugs, and then validating those with how that played out in the real world. An extraordinary acceleration of what typically used to take many years. Ewan, over to you. Tell us a bit more about the viral genome side of this.
Thanks. So if we go to the next slide, please. Hi, so first of all, really, I'd like to just kind of discuss really what the landscape looked like in the UK kind of pre-COVID, and really the viral and human genome sequencing in many ways was two separate camps. Pathogen genomes were generated by research institutes, academic institutions, public health agencies, and this data was all made public in resources like the European Nucleotide Archive. And analysis was often it's carried out on institutional computation or within shared resources like the CLIME system that I'll talk about in a little bit more detail. Likewise, human genomes generated by both private sector as well as research institutes and academic institutes, again made publicly available. And there was significant more work towards linking human genome data with electronic health record data, which is obviously critical for defining phenotypes of human disease. So we can move on to the next slide, please. So for those of you who don't know, sequencing of the SARS-CoV-2 virus really has been critical to the kind of global response to the virus. And really since January last year, sequencing has been ongoing. Sequencing is carried out on basically material that's taken from a nasal swab from an individual, where the RNA is extracted, and then the RNA is copied into a form of DNA called cDNA. And it's this cDNA that is then taken forward to be sequenced. And broadly, globally, most of the sequencing is done on one of two platforms. There's a platform called a MinION, which is produced by Oxford Nanopore, which is the size of a small mobile phone, or in much larger scale, it's often done on Illumina platforms. And within the UK, we've used both of these methods. So just move on to the next slide, please. So early in 2020, the COG UK, or the COVID-19 Genomics Consortium, was stood up in March 2020. And really within a couple of weeks, there were 17 different sites across the UK, including all 4 of the UK public health agencies, academic institutions across the country and the Wellcome Sanger Institute were all joined together to sequence the virus. And as I mentioned, within a couple of weeks of this being stood up, we were generating data. Broadly, COG UK operates by taking samples from both diagnostic testing in the NHS, and most of these samples flow out into the regional sequencing sites across the country, but now flow into, mainly in England, flow into Colindale in Public Health England. And then samples from community mass testing in the UK Lighthouse Labs networks, in majority flow into the Wellcome Sanger Institute that's turned around its high throughput pipelines to sequence. But all the data comes together in a single place in the MLC Climb platform, which basically allows us to analyze the data and conduct quality control, and to take that data and push it out into different places. The COG UK data then flows from this centralized system where it can be analysed and accessed by the public health agencies, flows out to the public health agencies where it's used on a daily basis for monitoring the spread of SARS-CoV-2 across the country. It's also made publicly available. Since day one, COG UK has made all the UK data publicly available with a limited amount of metadata. And this has been really important, obviously, for globally for other people to access and make use of the data. But as well as this data flowing into the public health agencies and into research, into the hands of researchers where it's been used, it also is starting to flow now into UK trusted research environments where there's been a huge amount of work to link it up with other kinds of data. So move to the next slide, please. So really over the last year, but really in the last 6 months, this has really gathered pace, working with colleagues from a wide range of institutions and organisations, and really spearheaded by Health Data Research UK. We've been working to link the viral sequencing data with other data sets. So we've been working with the genomic team to link viral sequencing data to human genomes, so that people can look at how particularly different viral variants may interact with different host genetics. And this again may have the ability to look at how susceptibility and outcomes are different between the virus interacting with the host. The data is now flowing into the Office of National Statistics, where viral variant information will be linked with demographic information, for example, to understand how transmission is going on across the country. And really, in the largest piece of work that we've been doing is that we're ensuring that the viral sequencing data can be linked en masse with the clinical outcomes, severity, comorbidity data that's held in a number of different data banks. So, for example, the English and Scottish viral sequencing data is flowing into the EPCC in Edinburgh, where it will be linked with the ISRIC cohort data about severity of disease. Again, it's also going into the SAIL data bank in Wales. And there's a large-scale effort now to routinely link viral sequencing data with clinical outcome. And I think in the UK, we're in a particularly good position to do this with NHS electronic health data. And the large-scale nature of the viral sequencing effort in the UK from early last year means that we have over 400,000 data points that we will be able to investigate questions like, do particular viral variants cause more severity of disease? And to do that, we obviously need to link to things like, do patients also have comorbidities? So this is work that's ongoing now, and hopefully there'll be some really exciting results that'll be coming for this over the next few months. So can we go on to the next slide, please? So what have we done to put the data that COG UK has generated over the last year to work already? What kind of questions have we already answered with it? So if we go to the next slide. So one of the first pieces of work that COG UK did was really looking at how the first wave of virus was introduced into the UK, and work really that was led by Andrew Rambo and Ollie Pybus, linked viral sequencing data to flight data, and could look where the importations of SARS-CoV came from in the first wave. And this identified that although a lot of the focus was obviously on China during the early part of the pandemic, actually the majority of cases in the UK, as you can see here in this figure on the left, came from Europe and came from France and Spain. So this provided us really critical information about the scale of the importations into the UK, and there was at least 1,200 independent importations in the first wave. We've also more latterly, some work that I've been involved in, we've been looking at how using genomic data to assess how well quarantine measures were effective in blocking transmission or importation, that is, over last summer. And this showed that there was a significant reduction in ongoing transmission by the quarantine that was required from people coming back from countries with no travel corridor. But the fact was that cases were still coming in. So this has provided important insights into non-pharmaceutical measures. So move on to the next slide, please. So as I mentioned, one of the really critical things that we can do linking to patient record data, and something that we're now hoping to do as a kind of a UK-wide scale, but that's been done much earlier on in the pandemic. This was work that was led by Eric Volz and colleagues, was looking at the D614G mutations. And this was one of the very early mutations in the spike protein that spread globally. And by linking to clinical record data, there were early reports that this may have caused greater clinical severity. By linking to the patient record data, they were able to show that actually there was no significant difference in clinical severity with this mutation, which of course was quite reassuring that there was significant worry at the time when this was first reported. On to the next slide, please. We've also been using the data since really last March to link to patient data in hospitals. So sequencing data has been used to track transmission. We've also had a large national study called HOKI, which has looked at the effectiveness of linking viral sequencing data to epidemiology in hospitals. And there's been some really fantastic examples of this used early on in the pandemic. For example, in Cambridge, sequencing was used to identify the source of transmission as actually patient transport. So measures were able to be put in quite quickly, rapidly reducing the likelihood of people acquiring infection. So that's been some of the things that we've been doing really with this viral sequencing data, and that's just a snapshot of the number of studies that have come out of COG UK. There's really been a huge research effort, and as well as feeding into the pandemic. Can we move on to my final slide now? So I think the thing that's really starting to emerge now is like, if we go back to our my first slide about that pre-pandemic, that we had quite a segmented system between pathogen genomics and host genomics and clinical record data. But I think now it's really, what's emerging is the need to draw all this together, that we need to have a kind of a integrated pathways for capturing of specimens and consenting patients to generate pathogen genomes, but also to generate human genomes, linking that with immunology data, biobanking samples, and bringing this all together in trusted research environments where this data can all be brought together. And I think one of the really positive things that will come out of what has been a tragic pandemic is that this kind of data linkage really is here to stay, and it's going to be an incredibly powerful tool for future pandemic preparedness, but also for infectious disease research. I've just gone to my last slide. And I'd just like to thank all the people who've been involved in the COG UK. It's been a phenomenal effort of over 600 people involved in on a day-to-day basis in the actual sequencing itself, but also all the colleagues in the Lighthouse Labs, NHS diagnostic laboratories as well, and test and trace. It's been a really huge, monumental effort, and I'd just like to thank them on their behalf. Thanks.
Fantastic. Thanks, Ewan. And it's fantastic. It's great to see the diversity of the team in the COG UK consortium there, including penguins, dogs, cats, and of course, Spider-Man. We know that diversity brings strength, and it is great to see that play out. So we've had some questions come through on the chat, and would love to turn to a couple of those. We have about 10 minutes before our sharp cutoff at 22. The first one I'm going to pick out is a kind of broad one, and I'll try and give Albert and Ewan a chance to think by asking Sue first, because I know she's hot on her feet. And if we could try and keep answers reasonably punchy and brief, that would mean we could get through more questions. So this first one is a reasonably broad one, which is, if you could keep one thing, or maybe one or two things, from the pandemic response that we can bring into the post-pandemic world from this kind of sphere, what would they be? Sue.
Thank you, Chris. I'm going to have three very quick points, if I may. The first is our ability to respond at speed with an end-to-end approach. That's both from the diagnostic to the data. And that's a critical, and we can be able to do that and respond at speed. The second is the alignment with clinical or public health data or with the research endeavour. And the third is that we do more in the host pathogen space over and above COVID-19.
Great. And Sue, maybe breaking my own rule, just say a tiny bit more about what that could look like for you, that final point, doing more in the host pathogen space.
So, for example, there's been, and the UK has led the way in TB sequencing. I've previously done work when I was actually a researcher on Haemophilus influenzae and a Haemophilus parainfluenzae sequencing and trying to find changes in the hosts, but not driven at that time by genomics. I think we've got a real opportunity with a number of infectious diseases to go back and start to look at the host genome in the way that genomics been able to do for SARS-CoV-2.
Super. And I'll go first, Albert, and then Ewan as well. Any brief thoughts on what we should keep or sustain from the response effort? Albert.
I think I agree with Sue. So if you could imagine a world where we would have had DNA or sequence for everybody, this would have been even faster. The question is how we get there, how we get people to consent to that. But I think it would be perfectly achievable. People actually give their DNA to companies for ancestry analysis. So why couldn't give them for health research?
For public health. Okay, we're gonna come back to that because it's a hot topic. Ewan, any thoughts?
Yeah, I think like Sue, I think the keeping the kind of the whole process in place, the ability to capture samples from the patient in terms of the pathogen itself, retaining that for the sequencing, and then linking that ultimately to data, and whether that be host genetic sequencing data or to clinical record data. I think the infrastructure that is in place basically for COVID-19 is perfect to be, with some modifications obviously, to be transferred over to routine normal business. And I think that if we could retain one thing, that would be an incredible, powerful thing to do.
Got it. And in fact, Ewan, I might stay with you and just kind of turn us back on ourselves. We've talked a lot through the presentation about linking these different sorts of data. You put up that great slide that showed a potential kind of future vision for how these elements can come together safely and productively and effectively. It was noteworthy that you had consent right at the middle of that flowchart, and also trusted research environments kind of off on the right-hand side. What do you think we can do both at a technical level, but arguably more challengingly at a kind of societal level, to provide the right kind of information, engagement, dialogue, reassurance, to maybe not quite get to the world that Albert outlined, where everyone has their genome sequenced and available on record, but to a world where we can really realize these benefits at scale from linking these different data sources together well.
I think we obviously have to advertise as much as possible the results of these kinds of studies. I suppose that in many ways that COVID-19 is going to be, over the next 2 to 3 years, there's going to be a great opportunity to explain how these individual components were joined together to understand. I think someone was saying the other day, I heard that we know more about COVID than we do flu, and how long has flu been around. I think that it's getting that message across, that how what's only been achievable through these, at pace, the development of vaccines, the understanding of the immune response, the understanding of host susceptibility, how trials have linked together with data. I think really exploring the possibilities of what's been developed during this pandemic is critical. But I also think that obviously the education of the general population is going to be critical to bring them along with these changes, and to understand how, by consenting to, if you have, a family member or you have a serious infectious disease, how that may help prevent others from going down the same path, I think is probably the critical message, isn't it?
Super. And Sue, maybe if I can come back to you, we had incredibly high levels of public support for the way that data was used in the crisis, and indeed we saw trust increasing for not only the NHS, which is typically a very high trust organization, but also for pharma companies and tech companies through the crisis, who had more recently been sort of lower trust organizations. Do you think we can sustain that into the post-pandemic world? What do we need to do to be trustworthy and to kind of earn that trust?
Yeah, so Chris, just very quickly, and linking with the Genome UK strategy, I think one of the elements we've got to do is continue the public dialogue. That's been recognised in Genome UK. Pathogen genomics is now part of it in terms of the implementation plan, but we've got to build and sustain and recognise public dialogue will be a key part. And that will take us also from consenting or informed choice right through to a greater understanding of data sharing for individual benefit, but also for population benefit across the world. Absolutely.
Okay, and so my final one is going to be a sort of one sentence answer for each of you, because we're just about right up on time. If you could think of one other area of disease where these kinds of approaches that we've talked about, linking the multi-omic data with the clinical data and other kind of public health data sets, which area are you most excited about the potential for? Sue, I'll start with you, then Albert, then Ewan. Sue, which other area can we apply these most effectively?
So Chris, although I didn't have time to go into this, we're already doing this for rare disease, for some common diseases and for cancer. And what I would say is we just need to accelerate that and make sure we cover all cancer and we move more into common diseases.
Super. Albert, your one sentence, where are you most excited about applying these techniques?
Common diseases, cancer, late onset diseases.
Super. And Ewan?
Back to my own research interest, bacterial infections, and particularly, I think, thinking about the antimicrobial resistance, this approach is going to be incredibly powerful.
Absolutely. I'm not sure if you're on Sally Davis's payroll, but I'm sure she would be pleased to hear that as well. We are right up on time. I can see our friend and colleague Natalie Banner here, who I suspect may be doing something in the next session. So we should probably hand over to them. But thank you, Professor Dame Sue Hill, Professor Albert Teneza, and Dr. Ewan Harrison for this talk and for your time. Thanks for everyone for coming along.
Thank you.